Welcome everybody to today's coffee seminar. Randy Bartels, Associate Professor of the Engineering Department at Colorado State University. Randy got his PhD degree from University of Michigan, but working here at GILA with part of the and then he joined the faculty at Colorado State. He received many recognitions and awards, among them the Adolph Lamb Medal from the Young Society of America, the Beckman Young Investigation Award, the IEEE Leos, now Photonic Society Young Investigation Award. He was named a Kavli Fellow of the National Academy of Sciences and received a Presidential Early Career Award for Science and Engineering in 2006. So, thanks for coming.
All right. Great. All right, well, thanks for having me. So I'll talk a little bit about one part of what I do in my research group up at CSU. And in general, so I've developed over the last sort of 4 or 5 years an interest in various sorts of optical microscopy, and we're essentially interested in 3 different aspects that I'll tell you a little bit about. One is moving to very high-speed 3D microscopy, and we have two different topics that are moving in that direction that I'll talk about here. We're interested in, like many people, techniques for improving spatial resolution. I'll tell you about an experiment where we looked at doing that with coherent nonlinear optical microscopy, third harmonic, and also very sensitive molecular detection. I'll tell you about some of our work on that in the past, and we have a lot of ongoing efforts along those lines right now. But before I get into this, as you guys know, because you're students, you guys are the ones that really do all the work, and I'm just a spokesperson. So the people who did the work that I'll tell you about today are David Winters, who's a student who will soon be defending in my group, Omid Mezadehi, I still can't pronounce his last name, who graduated about a year ago or so, Jesse Wilson, who graduated about 6 months ago, Philip Schloop, who was a postdoc and research scientist with me, Greg Fatia, and Dave Smith. And the work I'll talk about also involves collaborations with Stu Tobit, who's a developmental neurobiologist in biomedical sciences at Colorado State University, Jeff Squire down at Colorado School of Mines, and then Ali Pazeshki, who I forgot to highlight, is working on some extensions. We won't talk about any of the results there, but we're working on extensions of some of the things that I'm talking about here today, and I'll point that out later on. But that's probably particularly interesting because it essentially involves some computational algorithms relating to the field of compressed imaging. And of course, funding from various sources. Primarily what I'll talk about today has been funded by a combination of National Science Foundation, previously from the Beckman Foundation, NIH, and also recently Keck Foundation. So the roadmap is I'll tell you a little bit about nonlinear laser scanning microscopy. Many of you have probably already seen this, but I'll remind you of some of the virtues of this approach. And then we'll talk about just a couple of experiments that we've worked on. Functionalization of third harmonic microscopy, as I call it, essentially trying to get vibrational information, spectral information out of third harmonic microscope images. Enhance spatial resolution in third harmonic microscopy, so in trying to improve spatial resolution. Label-free second harmonic generation holography, with the goal there doing very high-speed 3D imaging with a nonlinear microscopy modality. And then a single pixel imaging that essentially we're packing one-dimensional information into a single detector by mapping spatial information to the frequency domain of the signal coming out of the detector. Okay, so one issue in various sorts of microscopies, when you talk about doing biological microscopy with optical techniques, is light is highly scattered by biological tissues. So you have limited penetration depth and light images get degraded. And this just goes with the typical Rayleigh scattering, where the strength of the scattering scales is 1 over lambda to the 4th. And just as a, sort of appreciate the issue here, so there's a simulation with just a Monte Carlo simulation of scattering media from Ming Gu's group from a number of years ago, but it shows, the degradation of a ring of light with propagation through larger and larger chunks of scattering media. And in this particular case, essentially you degrade your ability to see this ring certainly by beyond about 150 microns. And of course, the wavelength of the light that you use strongly depends on how quickly this gets degraded. So one way to deal with this is to push to longer wavelength light, and that's one of the motivations behind so-called multiphoton microscopy. So the idea of multiphoton microscopy is you send in light of one wavelength, and if it's intense enough, which means you require a focused femtosecond laser field, usually, you can stimulate various sorts of processes such as two-photon absorption or other nonlinearities that then localize any sort of signal generation to a small region near the focal volume of the light. It also means you're sending in light that's much longer wavelength than you normally would, which means you can penetrate much deeper into tissues. And so there are a lot of advantages to various sorts of nonlinear scanning microscopy. So like I said, you get localized information, so it gives you optical sectioning because you can constrain your signal generation to a small region near the focus. You get slightly better spatial resolution, although it's up to close to a factor 2. It's not super significant. The long wavelength excitation is extremely useful in scattering tissue, so it's very good for in vivo biological microscopy. And you just build up an image by the known location of a focus. So you scan things around. The only drawback with that is it's a relatively slow process. And for those of you who work with fluorescent microscopy, there's also the advantage that you're only exciting fluorophores. You're doing it through a nonlinear interaction, so you're only exciting fluorophores in a small focal region, so you're not causing photobleaching of all of the out-of-focus fluorophore molecules as well. But you also have, and this is what I'll focus on mostly, you have non-fluorescent contrast mechanisms, so you can generate new colors with multiple, with nonlinear mechanisms that aren't fluorescence-based, and you can get around some of these photobleaching issues. And this is what I'll talk about for most of the talk. So the two most common modalities for coherent nonlinear microscopy, or two of the most common, one is second harmonic generation. So for the physicists in the audience, you usually think of this in terms of an energy level diagram, and the basic idea, as you know, is you combine two low energy photons and you can scatter a high energy photon that's twice the input frequency or half the wavelength. And from symmetry considerations of this fundamental process, what you require are various sorts of molecules and structures organized in a way that they don't have inversion symmetry. So they look different one way compared to the other. So it turns out various sorts of structural proteins, such as collagen, myosin, tubulin, things like that are organized into various types of structures, such as extracellular matrix formed by collagen, various muscle fibers, and you can also have signal directly from membranes or dye molecules compartmentalized in membranes. You can get endogenous second harmonic signals from tissues directly that tend to be relatively strong, and they pick up only on these various structural tissues. So you get structural contrast out of these images. And this thing is not happy. There we are. Okay. So this, and the other thing about this is it turns out with second harmonic signals, particularly if you vary the input polarizations, you can get a quantitative measure of order. So you can get information on the atomic ordering and the ordering of the various structural molecules into fibers. And this turns out, this has been looked at in some detail over the past sort of 5 years or so, can be a nice reporter of various disease pathologies. So if you have muscle fibers that start becoming disordered. It could be indicative of some sort of muscular atrophy-related disease. And so there's lots of interesting clinical applications. It can also be used to look at dynamics. So here's an example of a relatively recent experiment out of Mark Schnitzer's lab at Stanford. And what he did was he took, and essentially, so if we look at this image here, this is an image we took in my lab of a skeletal muscle cross-section. So this is a second harmonic image, so the intensity just maps the strength of the second harmonic signal. And what you see are, so these sort of strands represent various muscle fibers, and these stripes are half of the sarcomere, which is the fundamental contractile unit of a muscle, and these are myosin 2 thick filaments in the sarcomer units. And so what Mark did in his lab is they in vivo they scanned one-dimensional line of some of these sarcomeres and then induced contraction in these muscles and watched the change in the unit, essentially the action of the contraction of these muscles that then correspond to changing these sarcomer lengths as they pull together. And the important part about this is first of all, this is just providing some one-dimensional information, and we're interested in developing techniques similar to this, but that let us see the three-dimensional organization at high imaging speeds. And if you take a look at this, on a tens of millisecond time scale, you see contraction and then relaxation of the muscle sarcomere lengths. And so if you want, and of course muscles are organized in biology in general, is organized in 3 dimensions. So we really want a 3-dimensional picture of these sorts of dynamics that are happening out of these sorts of endogenous structures. So that's second harmonic generation, so I'll talk a little bit about that. Third harmonic generation is, so I have my energy level diagram. So this is, instead of adding together 2 photons and scattering a higher energy photon at twice the input photon energy, we combine 3, hence third harmonic. And so we have light that's scattered at 3 times the input harmonic photon energy, or a third the wavelength. And it turns out that sort of fundamental work, when third harmonic was first observed, demonstrated that if we take any sort of light, CW light or short laser pulse, and we focus it really tightly in a homogeneous medium, then what happens is we generate third harmonic light all throughout a focus. So this represents the intensity of our focus. And it turns out that with third order interactions, we always generate a third order interaction. We're not symmetry limited like second order interactions. But if you put a detector over here under this situation, you measure no far field third harmonic scattering. The reason is that even though you're generating third harmonic at every single point within this laser focus, what happens is that the phase, the so-called Gouy phase shift through the focus, is an odd function, and it turns out that you get an exact cancellation of all the third harmonic generated before the focus with the light generated after the focus. So what that means is if you somehow disrupt that symmetry by putting, for example, an interface or a small particle within the focus, somewhere near the focus, then you will generate a far field third harmonic signal somewhere at the back end. And so here's for the physicists in the audience, there's our energy level diagram. The signal goes as the cube of the intensity rather than the square, so you get slightly better localization compared to second harmonic, which is not really that significant. The important thing is this gives interface contrast and lets you see very small objects that are small compared to the focus. So you can see various sorts of, you can see tissue interfaces. So this is just a picture of some epithelial cells, cultured epithelial cells that we took in my lab. And so you can see, essentially the cell membrane and the nuclear membrane there. The other thing that you can often see are various sorts of organelles that are small and various sorts of lipid vesicles that will show up. So there are 2 very different techniques that give you very different information. And the other thing is, as I said earlier, the way these images are built up, whether these are second harmonic, third harmonic, or 2-photon fluorescent microscopy, you focus your light to a single point, and then you scan that around, and you can build up a 2D image or a 3D image by scanning that around. And so to get an idea for how fast this goes. Typically, just because of signal considerations, you sit at one point for on the order of something like a microsecond to 10 microseconds, and you can scan out a 2D image on the order of a second or a large fraction of a second. And this is a relatively slow process, even in 2D, and then trying to move to 3D is even slower. So we'll talk about some ways to speed that up. But first, we'll talk about adding vibrational spectral information to third harmonic microscopy. So the idea here is, this is fundamentally related to pump probe type spectroscopies. So what we do is we come in with a laser pulse, so typically about 15 femtoseconds, and we focus that, we compress it so it's 15 femtoseconds at the focus of a microscope objective. And what that does is that excites coherent vibrational motion in the molecules. So essentially it's like hitting a bell and making it ring with all the vibrational modes that are shorter than how quickly you hit the bell. So we can excite vibrational degrees of freedom that are Raman active with vibrational periods, if it's a 15 femtosecond pulse, that are longer than 15 femtoseconds. And so the way we think about this is we say, well, okay, if I think about what happens, not only do I don't typically have one molecule there, I'll have a bunch of molecules there. And when you ping them, you essentially get them all vibrating in phase. It's kind of like line dancing rather than a mosh pit. And so they're all sort of dancing around in sync. And what that does is that changes their optical properties in a relatively large way that we can detect. So you can get a change in the effective linear susceptibility, which basically means that there's a change in index of refraction or a phase shift that follows. And that's what this wiggly wake represents. These things start bouncing into each other and they dephase. And so the strength of their line dancing, they go from line dance to mosh pit eventually. And if I come with a pulse later on, I can interact with that and then pick up various sorts of optical properties. So we can measure changes in the optical, in the index of refraction with some sort of interferometry technique, or we can measure changes in third order susceptibility. And to get vibrational information, we need to scan a probe pulse, after that initial excitation. So this time here, so this represents time in a delayed probe pulse, and we would imagine we would have a pump pulse exciting the molecules right here. And then what we do is, it's just essentially time resolved ultrafast vibrational spectroscopy in the end. But we read this out in a microscopy format. And ultimately what we're doing is we're looking at modulations in the third harmonic generation signal that we collect. We scan that delay and we give vibrational information. So the experiment looks basically like this. So the first order version of this that we did, we started with crystal interfaces, so we would excite optical phonon modes in the crystals, and then scan, we focus a pump pulse, scan a probe pulse, and record the third harmonic signal, and then resolve those vibrations, Fourier transform that, and get the Raman spectrum. So it turns out this is the low frequency phonon spectrum of a scintillator crystal BGO. So the basic idea here is, what we were after in part is being able to map out, image structure and pull out, or spatial third harmonic images. So we can scan this around in principle, and then pull out, vibrational structure information locally at each point simultaneously. The problem with this approach is this was a slow scan in the way we initially did it, and this took several minutes. So, taking several minutes to take one data point and build an image is absolutely useless. So we looked at ways to speed this up. And also the signal was relatively weak in this case. We had these cascaded lock-ins, and so this was a cumbersome experiment. So what we did is try to look for a relatively simple way to speed this up. So we were just using stepper motors. And so what we did is what we call the lighthouse scanning technique, because this is a really dangerous experiment where we send in our laser pulses into a birefringent crystal, slap it on a motor. We started with taping it to a $3 motor, spinning it around, and we get a beam that spins all around the lab, so we need to box it up carefully. But the idea is that we come in and so I'm sure you know about birefringent crystals that, there are certain degrees of freedom where if you send in, if a uniaxial birefringent crystal, if you send in a linear polarization along two special directions, your polarization stays linear, your ordinary and extraordinary eigenpolarizations of the crystal. And if you come in with a laser pulse that excites a mixture of those, we just use an equal projection, it turns out then if you cut the crystal in the right way, as you rotate the crystal, you'll get a variation in the group velocity, the transit time for, or the group delay, the transit time for the extraordinary polarization. It becomes dependent on this angle. So what we can do is when we spin this thing, we're rapidly scanning the relative delay between these two pulses. So we can, if we dump more energy in one of these, that serves as our pump, less energy in the other, then serves as our probe, and these can be scanned really, really quickly. So we come in, we put in a crystal beforehand so we can get time zero overlap, bounces off a mirror, come back, and essentially go into our normal experiment. And so we replace our $3 motor from with a $200 frequency stabilized motor so we could get a little better data. And then we went from something that took several minutes and averaging to get a decent vibrational spectra to get something that is essentially vibrational spectroscopy on a real-time scope. And we could update this at several hundred hertz. And it's a simple approach. And when I first thought about trying this, I had a list of about 20 reasons why this wasn't going to work. Primarily, my biggest concern, incidentally, is we spin this thing around, we have huge variation in Fresnel reflections, and I thought that alone was going to kill us. And it turns out that wasn't a big deal. Of course, I didn't tell the grad student my list of reasons why it wouldn't work, and he got it working in a day or two, and I was shocked at how well it worked. And then we could get really nice spectra on various sorts of things. And so the next step is to look at this for microscopy. We've been working with a chemistry colleague to look at to probe the structure of molecular monolayers at interfaces, with ultimate application to looking at charge transfer reactions for dye-sensitized solar cells. So we're doing more chemical spectroscopy applications of this, but eventually we'll probably come back to microscopy. But essentially, what this lets us do, and I also don't have the data, but you can separate out the contributions of the signal from interface contributions and bulk contributions with this technique by taking a set of data as you scan your interface through the focus. So it turns out to be a unique probe simultaneously of both interface vibrational structure and bulk structure. And so we're also hoping that we can do some studies of vibrational energy transport across interfaces and into the bulk of materials ultimately. Okay, so that's one technique where we're looking at molecular detection. That's not the downside of this is it's not super sensitive, and we have projects, a new project where we're trying to do very sensitive detection. So the other thing I mentioned is, we're interested in improving spatial resolution, as many people are, and we wanted to look at ways that we could do this with coherent nonlinear microscopies. And so the basic point to remember. So what do we mean by resolution in microscopy? What we mean is the ability to differentiate 2 points, say within, that are closely spaced to one another. And normally we worry about the point spread function, and if those 2 points are separated by a distance that's on the order of the width of the point spread function, we say we can resolve those. So, one approach, which is ultimately borrowed from STED, which I think many of you are familiar with, is you use some sort of nonlinear depletion mechanism, for example, to reduce the effective spot size of fluorescence emission. So we sort of borrowed that generic approach and did a similar sort of thing with third harmonic generation microscopy. So the idea is that we have a focus field, and we control the polarization state across that focal field. And it turns out when you look into some details of nonlinear optics, if you have linearly polarized light, you get strong third harmonic scattering. If you have circularly polarized light for isotropic media, you get your third harmonic scattering is identically quenched, or not quenched, but it's identically zero, so you get no scattering. So the simple idea is say, well, okay, we can take two orthogonal linear polarizations, and one of those beams have a flat phase, on the other one put a phase jump of pi over 2 at some radius, and we could switch, conceptually from linear polarization at the center of the beam to circularly polarized at the edge. And then the idea is, once you go through some third harmonic generation medium, instead of generating third harmonic light over the entire spatial distribution of your focus field, you have it confined to a narrower spatial region. Okay. So first thing, and so if you have an isotropic medium, this is what your third order polarization density looks like, which is proportional to the strength of the third harmonic you're going to record in the far field. So what we did is we wanted to characterize the difference between linear polarization and circular polarization. So we just made a sample with a small nanosphere, about 100 nanometers in diameter, use that as our third harmonic source to profile, essentially profile the point spread function of our third harmonic beam. So if we go to linearly polarized light, we map out a nice point spread function from our focus at our microscope objective. This is just with galvo scanning, laser scanning microscopy. And now we just put in a quarter wave plate to make that polarization circularly polarized, and we get no signals. This is just a noise pattern. So the fundamental idea, if we can switch the polarization from being linear at the center to circular at the edges, then we should be able to see really high contrast in suppression of that third harmonic generation. So to control the polarization state, what we used was a spatial light modulator from Boulder Nonlinear Systems, which many of you are familiar with. This was a relic from some earlier pulse shaping experiments, so it was a 1D SLM. We would have liked to have done this in 2D, but this was good for proof of principle. And essentially the idea here is we go through some galvos, but to first order we take this plane on the SLM and we image that, demagnify that to the focal plane of our objective. And the way SLMs work, essentially you have one polarization that comes in where you can shift the phase of that through rotation of your linear liquid crystal models, molecules, and then an orthogonal polarization, if it's say horizontally polarized would be affected by the modulator, vertical would not. So we come in with a mixture of amplitude, broken up between vertical, horizontal, and vertical. So we can impose a phase shift spatially to different parts of the beam. So if we introduce a pi over phase shift on the wings of the beam, that part of the beam becomes circularly polarized. That then gets mapped to the focal plane of our microscope system. So we just scanned around the laser beam with a small probe beam and took a look at what happened. So we went about characterizing the point spread function. I guess I should have had this slide earlier. So essentially we put this together and here's what happened. So what I'm going to show you is, so this top hat represents the phase profile that we put on the SLM. So it's all one-dimensional. So this vertical dimension is going to be the dimension where you see changes in the beam width that we map out, and then this sort of horizontal x dimension is going to be unchanged because we use a 1D SLM. So the nice thing about that is we have an intrinsic comparison, although 2D would have been better. And then we'll just show kind of the integrated energy, third harmonic energy, and also the improvement in the width along that y direction. So we'll just see the phase step come in from the edges, and we'll see the beam size shrink correspondingly. And so you can see that basically it works, and then at some point we just get a decrease in the signal level, and that's essentially because the whole beam, when this top hat becomes really narrow, the whole beam is approximately circularly polarized, and we just lose all of our signal. So this experiment actually went together really quickly. Then we spent about a year trying to figure out and perfect being able to probe the inhomogeneous polarization state. It's kind of pointless to publish a paper showing this if we can't show that it works for the reason we said it does. And so we were working in a moderate NA regime. So we done, it's for those of you who know, microscopy well, you know that if you have very high numerical aperture focusing, you start developing a significant longitudinal field. You essentially get a distortion of your input polarization at the back focal plane of the objective, and it's not preserved at the focus. If we restrict ourselves to moderate NA, it's not too bad. So that's our tacit assumption in all this, in order to then just do relatively straightforward probing of the polarization state with, again, our small nanoparticles at the focus. And so what we did is we just put an analyzing polarizer at the back focal plane of this objective, so we could then get linear polarization projection along one direction, get an orthogonal projection, and then mix it at some angle in between, typically 45 degrees. And so the basic idea there, it's essentially typical ellipsometry type measurements, which essentially work something like you have a total field, you decompose that into some x and y spatial distributions, and what you really need to, the thing you want to get out of that is the relative phase information, and it's sufficient to take a projection along two orthogonal directions, something that mixes those, and then you can easily reconstruct what the phase difference is. But the problem was that these measurements weren't linear. We were measuring a third harmonic signal, but it turns out that you can still find, you can solve this equation, and if you end up generating a third harmonic signal, you end up with 3 solutions instead of 2. 2 of them are complex. One is real, and the real solution turns out to give you this relative phase information. So by doing the same sorts of things, assuming the polarization is preserved through the tight focus, you can then work out the polarization state distribution at the focus of your microscope. But one of the other problems that you have to deal with is, so you take and you prepare your probes, which you want, ideally a small isolated nanoparticle, but these things, when you spin coat them onto some sort of cover slip or slide, they can tend to cluster. So one of the things that took the student me a lot of time in the laboratory was to find a particle that behaved isotropically. So if you have a cluster of particles and you go and you spin your half wave plate to scanning the polarization, if it's not, if your response of the system isn't flat with spinning that half wave plate, for example, that means that what you have is a cluster. You don't have isotropic behavior in the system, so it's not a good probe of your polarization because it's non-isotropic. So there's a lot of hunting around, even with, you have to prepare these things relatively dilute, spin them on, and still hunt around for a while to find one that behaves isotropically. But if it's isotropic, then your third harmonic signal, it should be basically flat by rotating the half-wave plate and be suppressed when you have circular polarization. So this is the sort of thing that we were looking for. Then you could go in and with a simple algorithm, go in and put on. So what we did is put on a specific phase change that's uniform across the beam, and then go make sure we could recover that. And so you can see, if we follow the line that we program on, our data should land on that. And so it basically works. And what you see is higher signal to noise over here on the right, and that's simply because we used X polarization as our reference. And when that starts getting small, when it goes to Y polarized, then you have a lot of noise. So if we switch our reference, this signal to noise will come down. So anyway, it basically works. And then the bottom line story is that if we go in and take a look at, map out what our polarization state is. So here's, for example, our shrunken beam along the Y direction. And we could go in and then recover what the polarization state distribution was across here. And what we found was, indeed, we had a linear polarization at the peak of the beam, and it was circularly polarized right here. And then we get a recovery over here on the edges. And that's because, we put a very sharp phase profile at the SLM, but then that gets, re-imaged and demagnified in the imaging system. But, the diffraction doesn't capture all those spatial frequencies, so this just gets essentially rounded off. So it's no longer top hat. There's some diffraction ringing. That's why we get a recovery of the third harmonic on the edges. And if we take our polarization state distribution and compute what we expect for the image, we get the same thing. And we have a color map. We have an image that has a matching color map somewhere, but I don't know where it went. Anyway, so we're able to show that we get good correspondence between what we expect with a measured polarization state distribution of the focus, and it's killing off the third harmonic on the edges of the beam as we expect. So what's the bottom line? We ended up getting about a factor 2 improvement in the spatial resolution. And we haven't done any detailed theory on this yet, but I suspect we may be limited here. And the basic reasoning is a relatively simple intuitive argument that if we think about, if this were a linear interference that we're giving a null in the beam, that would require about that would require a pi phase shift and equal amplitudes to get destructive interference and have a null. Now if we go to the third harmonic, well, we need to go from linear polarization to circular. That takes about a pi over 2 phase shift, and so it's about half the phase shift. So very naively, the factor 2 in improvement in spatial resolution, my gut feeling is that's probably a limit, so we haven't spent a lot of time trying to see if we can get below that, unless we can think of some method to sort of saturate that somehow squeeze that polarization tighter, the region of circular polarization tighter. Then I think this is probably going to be the limit. But still, a factor 2 in resolution is not that bad. Okay, so the next topic that I'll talk about is high-speed second harmonic holographic microscopy. And so here, again, back to the basic, normal nonlinear microscopy is a laser scanning microscope, and there are lots of good reasons to do that, particularly if you want to image deep into tissue. But the problem is it tends to be slow. So you do a 2D raster scan that takes on the order of a second, and then you need to move your focus, and then do another 2D raster scan, and that takes about a second. And it takes a while to move between those planes, although there's now a technique where you can move that in tens of microseconds. But even with that, if you want to do 20, 30 stacks, it takes several minutes to build up a three-dimensional image. And so, I already sort of talked about these structural, the contrast mechanisms, but essentially the laser scanning beam microscopy is slow. And so we're interested in very high-speed 3D acquisition. So why are we interested in that? For one thing, we want, instead of two-dimensional information, we want three-dimensional information because biology is organized in 3 dimensions. And there are lots of biological functions that rely on fast dynamic processes. We talked about some of those already, but generically various sorts of excitable cells like neurons. So if you want to understand neural processing at a neural circuitry level, you want to look at a collection of 100 or 500 cells. You want cellular resolution or maybe even subcellular resolution, and you want to be able to look over a cube, at least a width of say 100 microns or so, or more. And the dynamics are fast, so the sort of width of a typical action potential spike is on the order of a millisecond. So what you'd really like to be able to do is if one cell gets excited, you want to see how it's connected in space and time to other cells. Another application where there's been a big push to do high-speed imaging is looking at embryonic cardiac dynamics. There are various sorts of models for embryonic heart development in an effort to understand congenital heart defects. And a lot of these model organisms have heartbeats that are well over 100 beats per minute. So that means if you want to be able to resolve the 3D structure of the heart dynamics, you need to be able to image at rates of many tens of volumes, cubes, per second. So these are two motivating applications for high-speed imaging. And so one way to do high-speed 3D imaging is off-axis holography. This was developed by Emmett Leith. And in the late 60s and published in the early 60s and later. Anyway, the basic idea is you take light and you sort of coherently mix it with some light coming from the object, in this case reflected, with a reference, and you record that on some sort of two-dimensional imaging format. And it turns out that that gives you three-dimensional information. And the way I like to think about this is if I think about different points on the object and say I'm getting, think of those as individual source points or scatter points in the object. And then if I come over here on some sort of 2D format detector, record a two-dimensional image, it turns out that when I look at the interference between, say one of these points and that incident light, I record fringes that turn out to be a Fresnel zone plate lens pattern. And the focal length of that Fresnel zone plate lens tells me about the distance from that recording plane back to my object. So in that interference pattern, that ring-structured interference pattern, what you end up is encoding depth information. Of course, the relative amplitude of each of these points gives you amplitude information. So you can get complex amplitude and phase information from a single snapshot image. So that's the real value of 3D off-axis holography. And then it's very straightforward in order to reconstruct that. And so, of course, holography was initially developed in electron microscopy context by Gabor. And essentially this idea of thinking about holography as a superposition of Fresnel zone plate lenses was due to Rogers a couple of years later. So then the scheme is to do this with nonlinear microscopy. And so this is a little more challenging. So this was developed by Dimitri Soltis's group. Well, he was around the time he was moving from Caltech to the EPFL in Switzerland, the second harmonic microscopy. And so the big issue there is you need pretty energetic ultrafast laser beams in order to generate sufficient second harmonic signal from a sample, because instead of now focusing to a very tiny spot to generate your signal, you have a much larger beam that you very loosely focus into your sample, so it's millimeters wide instead of a fraction of a micron wide. So your intensity has gone down significantly, so you need higher energy pulses in order to do that. But then, inside of interaction with the sample, so what I have labeled here is green structure that is second harmonically active. The red stuff is not. We come in with pulses that are red, it turns the light into green light, and so that represents these various wavefronts coming from the second harmonic scattering in the sample. And then you bring in a coherent reference beam. And because this nonlinear interaction for the second harmonic is coherent, you preserve your coherence with your external reference beam, and you can record holograms. And then, what we really want to be able to do, if we want to be able to nail three-dimensional neurological circuits, we want to be able to take a snapshot of this, 2 dimensions, that gives us three-dimensional information every millisecond. And so that's why our counter increments there. And then once you record those, I already said that, once you record those, and then you numerically reconstruct with just simple Fresnel propagators the three-dimensional information. And you can get a three-dimensional movies as these move along. Numerically back propagate. Running out of time, so I'm going to skip some details no one cares about. So the basic setup looks something like this. So we bring in our pulses, and so these are pulses from a ytterbium YAG laser that's reasonably high average power. Split off some of that, generate a reference arm, go through the sample, and then collect the light, and then send it onto some sort of camera. And then to process these, we do this numerically. So very briefly, we take the raw hologram, do a 2D Fourier transform, and then it turns out one of these sidebands gives you the complex field information. Once you have the amplitude and phase, that's sufficient information to be able to back propagate. So you isolate one of those fields, use numerical propagation, and you can reconstruct a three-dimensional image. And it turns out that you can get good, essentially numerically derived optical sectioning that's dependent on the numerical aperture of the hologram you collect, and you can back propagate that over an extended length, so that gives you an effective sort of extended depth of focus with this sort of technique. And so what this looks like, so here, let's let it go. So here on the left, this is a measured hologram from 3 starch granules. They're potato starch, and I need to have them not zoom over such a wide range. But anyway, you can see over a range of back propagation distances, I don't know what scale this is. I don't think it's calibrated. But you can see reconstruction of at 3 different locations, these starch granules. So you get 3-dimensional information there. So the advantages of second monocolography, one, you get you're mixing a relatively weak signal with a strong reference signal, so you get a boost in essentially your sensitivity through heterodyning. This is numerical optical sectioning and correlated extended depth of focus that I told you about. Also, you have interferences polarization dependent, so you can select out different polarization components of the nonlinear interaction tensor and get more information about the samples. And our big interest is high-speed 3D imaging. So here, and you can get this from endogenous tissue samples. So this is an example of muscle fiber. So this was taken with one snapshot of a hologram. Then we numerically backpropagate various distances. And so I showed you some skeletal muscle earlier, and you saw these sort of sarcomere structures organized into fibers along this direction. And this is just a isosurface rendering of constant second harmonic signal strength. And just to show you some of the polarization dependence, is if you vary, for example, the input polarization, you can pick out different structures which correspond primarily to different alignments of these muscle fibers within that interaction volume. And so we're working on analysis of some polarization resolved holographic data to be able to get orientation of the fiber structure inside of these. And the important thing is that we can get down to a millisecond exposure time, and we can do that at a continuous rep rate. And so I couldn't find one of those images this morning, but we have now images of up to 1,000 frames per second. So in other words, we can get a 3D volume updated every millisecond of starch granules, which granted are a very strong second harmonic emitter, but we can see them move around in 3 dimensions. So the downside of holographic microscopy is now we're susceptible to scattering. So the fact that, so we come in, for example, so we're looking at various tissue slices, and we're working with this Stu Tobit, a developmental neurobiologist, to look at various tissue slice organ models. And so the issue is that, so what I'll show you is some data where we tried to go deeper and deeper into the tissue. So for various technical reasons, the range over which we can reconstruct the holograms is limited, anywhere from, depending on the setup and numerical considerations, 30 to 100 microns at this point. So we can get a reconstruction over some finite range. So what we can do is align things so that we have a reference plane somewhere and reconstruct it in some region around that. And so here we're showing, so what we have is our fundamental pulse comes in from the back of the tissue, and then we collect the light from the front, and then so this is an image of a fixed skeletal muscle tissue, 250 microns thick, and we're collecting signal from near the top, and we backpropagate that 30 microns in. Now the problem is that when we generate the second harmonic light early, deeper inside the tissue, there's lots of scattering that happens on the way out. So what you would expect is an image degradation. And so this is, say, 60 microns deep. And so on the left we have a skeletal muscle reconstruction, the right cardiac muscle. So you can still see pretty reasonable reconstruction of this. And we have slices that go deeper and deeper, but you start getting degradation of this as you go in. So this is one of the issues with this technique. It's ultimately a wide field microscopy technique that suffers from scattering, although Dimitri Soltis' group has demonstrated you can do some optical conjugation to mitigate this. So, to do tissue slice models of 3D imaging at high speed, I think would probably require that sort of phase conjugation implementation. Okay, so I'm almost out of time, probably out of time. Am I out of time? 5 minutes, okay, I'll do a 5-minute version of this. Okay, so the last topic I'll talk about what is I wanted to come up with an imaging technique with a good acronym. So I like to say my images are spiffy. So here we'll talk about spatially integrated chirp frequency modulation imaging, and I selectively took some of the letters and called it spiffy imaging. But the idea is actually simpler than the name. And the idea is relatively simple. So the idea was, okay, so I just told you about this holography, which is ultimately a wide field technique, and it's susceptible to scattering. And a good way to get around scattering issues is use a single element detector. So, but then it's slow, so this is an approach to try to speed up an imaging technique that can work in a scattering environment, so in a turbid media. So the idea is we want to somehow map onto a single detector spatial information. So we take a laser beam, focus it to a line focus, and then we build a modulator that gives us a chirped intensity modulation frequency across the beam. So say on the top, here we have a low frequency modulation, and it gets linearly ramped in frequency, higher and higher frequency, to the bottom of the beam. And the idea then is that if we go through an object, we then transfer spatial information onto this modulated beam. We send that onto a single element detector, look at the spectrum of the signal, of the electronic signal coming off that detector, and we get spatial information. So the question is, how do we go about doing this modulation? And so we wanted a relatively simple way to test this idea. So we decided to go start printing on blank CDs, and what we did was look at patterns that essentially at each radial spatial point, so if we imagine a line focus, say along here, each radial spatial point would give us a different number of alternations between light and dark as this disk spun. So in other words, each radial position would give us a different intensity modulation frequency. We can jack this up and write down a mathematical form for this, and it looks like this. And then we can go and spin this disk and put a line focus on here and spin it. And then each point on this illumination beam sees a different modulation frequency, and we don't care about details. And then we go and we just send this into a DAC, do a Fourier transform, look at the at the modulation sideband, and we see a mapping of that spatial beam. So the optical setup looks something like this. We just take a beam, and this was nice because this was the first experiment I ever published on non-femtosecond laser pulses. Much easier lasers to work with. So we just took a CW laser beam, focused it onto a line, and then used some relay imaging optics just so we could put our object in a region over here, away from where our mask was. And then we just collected that light and sent it onto a detector. That's weird. I thought I had, no. And so here's what the signals look like coming out of the detector. So I had little unmodulated regions that serve as synchronization timing signals. That's these little squares of light. And then this is what the signal just from an illumination beam looks like. And this is a log scale of the spectrum coming off of there. So we can go and scan a single slit through here to characterize this. And so the spectral width of this slit was about 75 hertz, which, when we map this sort of frequency modulation chirp, corresponds to about 140 micron resolution, which is what we expected from calculations. And then we see the intensity modulation we map from the sort of spiffy measurement maps exactly what we get from the beam independent beam profiling. So everything seems to fit. And another way to think about this is we have a swept illumination of spatial frequencies through here. So at each different time point as we spin this disk, we have a different spatial frequency modulation. So if we think about that in the time domain, what we should then get is at each time sample of our measurement, we get part of the spatial frequency, the spatial spectrum of whatever object, the product of the object and the beam that we're sending through there. And that's also what we'd expect if we think, well, if our spatial information ends up in the spectrum of the signal coming out of there, time domain should be some representation of the spatial frequency representation of the object. So this is one way to think about that. The other thing about this is this is a nice, convenient way to think about diffraction of this beam. So essentially what we have is at each point we have a different spatial frequency of the grating, and so provided the numerical aperture of our collection imaging optics captures the highest spatial frequency diffracted, we're not going to get any distortion at the imaging plane from this system. But if we go and we think about, okay, if we move away from that imaging plane and we get defocus, what's going to happen is we start we have an aberrated, or a defocused point spread function. We'd start blurring out the highest spatial frequency content. And so the other thing we would expect from this is we might start getting sectioning behavior is essentially like structured illumination, except instead of sending in fixed spatial frequencies and moving them side to side, we now sweep the spatial frequencies. So we have some preliminary data that this is relatively new data that we haven't analyzed in detail, but it shows we can essentially localize our optical signal very locally, and we get better localization if we block some of the low spatial frequencies, because these low spatial frequencies are essentially constant; they never blur out. So that's just the background. That's this background here. So we're now exploring the capabilities of optical sectioning and how this behaves under scattering conditions. I'm going to skip this because we're out of time, although they're cool movies. So we can go and we can measure the standard Air Force test pattern, and we had a finite width of the imaging, so we did this in 3 stripes and just scanned the line up and down. And we can also go do this with fluorescence. The modulation frequencies that we're using here are in acoustic frequencies, so much slower, or much, with the modulation speed that's very slow compared to the relaxation time of fluorophores, which means that as we vary this excitation amplitude, the fluorescence emission adiabatically follows that modulation, so we can transfer this modulation information not only into the amplitude of the excitation, but also the fluorescence. Then we had to figure out how to build ourselves a fluorescent object. So then we realized we had some Sharpie fluorescent highlighters around, and we squiggled them on some slides, and the pink and the orange actually gave us a nice spectrum that matched the filter we had in the lab. And we needed an object, so I went to Hobby Lobby and bought the Spring Collection barrel of stamps. And we stamped that onto a slide, and that still didn't work that well. It turns out if you spray some hairspray and rough up the slide a little bit, you get much better adhesion. And so we can make ourselves little fluorescent objects, and then you can compare fluorescent spiffy in its fluorescent emission mode and its absorption mode. And here bright intensity is inverted, so darker here means lower light intensity corresponding to more absorption, and darker heat on the fluorescent side just means brighter fluorescent emission. And if so this is our crude dye transfer process here, made for very splotchy test images, but we could identify reasonably constant fluorescent and absorption regions of interest and compare them to the background, and you see what you would roughly expect, which is that the fluorescent image has about an order of magnitude less signal to noise. So they're nicer images just because you don't have a lot of shot noise from that background intensity like you do in the absorption. So one thing to remember is that we're integrating all of the light onto a single detector. So any light that's hanging around is contributing to shot noise. So fluorescence gives much better images than the absorption. And I am out of time, so I'm going to skip the last bit. So I'll just leave it at that, I think. Unless people want to hear another 5 minutes, but I doubt it. Probably had enough of me. All right, I'll just end it there. Thanks. Trying to just go to the conclusions. And if there are any students looking for jobs, I have a couple of postdoc positions open. That's my most important slide. Yeah.
So going back to way to that kind of the beginning, there was this slide that you had from Stanford that they did of the muscle tissue, and they had stressed it.
Yeah.
How did they do that? Is it a live sample or?
Yeah, that was in vivo with human test subjects, and I think what they did actually is just have the people flex their arm.
How did they image? Is it?
So it was so they had essentially an endoscopic grin lens that they poked in there,
Gotcha.
and then they would just scan along one dimension. And so with a line scan, you can update that every 2 milliseconds, roughly, and so they could get fast dynamics with that.
Underneath it for the layers of skin. Okay.
Right. So they had to find someone that they could poke with a thin chunk of glass.
You mentioned that the sticky approach is less susceptible to scatter. Can you talk a bit more about
Yeah, so, so are you familiar with confocal microscopies? Yeah, so essentially, as soon as so if you go onto a CCD and you have, say imagine you have a single region of fluorescence embedded in something that scatters optically a lot, and you excite that and you collect that onto a CCD. What you're going to get is your fluorescent spot and a fuzzy haze around there from all the scattering. And so if you think about, well, if you could just image, so suppose your dot were imaged onto a single one of those pixels and you only picked up that pixel, you're not picking up all that haze that's clouding the rest of the CCD image, and so you're rejecting a lot of that scattered light. So that's the simple version of it, if that makes sense.
That's good.
So in this holographic spectroscopic microscopy, what sort of energy level?
Let's see. So we have. It's been a while since I've looked at those numbers. So the oscillator. It's a ytterbium oscillator, putting out typically about 1.25 watts average power, and then we have to turn down the power going into the sample is quite low. Otherwise we blow things up and kill it, and so I'd say a majority of the power, probably well over a watt, is going into well over a watt is going into the generation of the reference arm, and maybe a couple hundred milliwatts is going into the object arm. And it turns out that I don't remember the number off the top of my head, but our peak intensity numbers, for our very loose focus with our condenser, is very similar to the tight focusing conditions for laser scanning microscopy, which isn't surprising because they crank up the power as much as they can until they kill things. So